Good morning, everybody. I'm Matt Hadfield. I'm a current third-year medical oncology fellow at Brown University in Providence, Rhode Island, and soon to be an attending focusing in phase one early drug development. Super excited to have this conversation today with Dr. Ahmadi and Dr. Nielsen of Genmab and AbbVie, as well as my colleague, Dr. Taneja. I think just to get it started, maybe we could start with just some introductions.
Sure. I'm Jackie Nielsen. I'm the Therapeutic Area Head for Hematology at AbbVie. PhD by training, trained at Northwestern Medical School in Chicago here. I've been here for 25 years or so. And yeah, worked on a lot of molecules in hematology, but so far, epcoritamab is my favorite.
Alrighty, Tahi Ahmadi. I'm the Chief Medical Officer at Genmab. I'm an MD PhD from Germany. I originally did my training on a follicular deal. It was kind of like one of the quintessential leaders in the lymphoma field. Then came to the States to do my postdoc in Boston, and then ended up doing my training at Penn, and then later on staying on faculty at Penn as an oncologist, where I was actually seeing mainly patients with lymphoma and CLL before I went into the industry. Initially to J&J, where I was involved in a number of drugs that are approved now. Namely Imbruvica, which I led the first approval, Daratumumab, which I led the entire development, Amivantamab, which I led the discovery, Tacrolimus, which I led the discovery, Tivdak at Genmab, where I'm now for 7 years, and now EPCO.
That's great. I'm Alankrita. I'm one of the third-year fellows at Roswell Park. I'm also going to be doubly trained in hematology and oncology. I have also signed up for a job offer to lead a myeloma program in Jefferson's Einstein in Philadelphia.
So you're coming to Philly.
Congratulations.
All righty. Congratulations. You're going to be at the same institution that my assistant was. Very good.
Thank you. I'm really looking forward to this conversation.
So I guess to get started, a lot of interesting things to talk about today, but maybe we could just start with talking about Epcoritamab, the mechanism of action, obviously a novel compound that works a little bit differently than immunotherapeutic agents in the past, with some very interesting details. So maybe we could just start with hearing a little bit about that.
Epcoritamab is a bispecific antibody. It's on the Genmab DuoBody platform, which has some unique features that might be important. But broadly speaking, the idea is to create antibody structures that mimic the behavior of an IgG1, but have FABs against two different targets. In this particular case, one arm of the antibody binds CD3, which is on T cells, and the other one binds CD20, which is on B cells. And by having these two arms, what you end up doing is you are bringing in close proximity a T cells to a B cell. And in the case of patients with lymphoma, this is predominantly their tumor. The antibody itself is Fc silent, meaning it doesn't have any Fc-mediated functions, which is important. And by creating proximity, you essentially trigger a reaction that frankly ought not to happen, but still does happen, and that the T cell then starts killing the B cell. So it's a T cell redirector, as there are many, many others in development and approved now. It's a relatively new space, I think triggered by the recognition that T cells have a profound ability to kill hematologic cells, and it's a very exciting area. What is unique about epcoritamab is beyond the fact that it's the one that the most closest follows the IgG physiology, meaning that it really looks like an IgG molecule, which is a function of the DuoBody platform, is also that it's the first one that was introduced subcutaneously. And the reason for that was the observation in the early non-clinical safety models that we did, that the peak PK would be modulated in a way that you would avoid it. And that was important to us at that point because we were working from the hypothesis, which ended up being true, that peak PK was what was driving high cytokine release levels, which is the predominant safety issue with this mechanism of action.
And interestingly, so it is such a novel drug we're starting to see more and more T cell engagers being used, bispecific antibodies being used, and particularly this is a subcutaneous formulation, which in the past we've seen things like rituximab go from infusional to subcutaneous. But have you noticed and we're going to talk about this more in depth, the phase 1/2 study, but have you noticed any novel toxicities that you would attribute either to the mechanism of action or the delivery mechanism that hasn't been seen with other similar molecules?
I guess the short answer is no. I think in the way drug development records therapeutic or treatment related or adverse events is or treatment margin adverse events is anybody that gets anything during the course of the trial it's recorded. So you get things like upper respiratory tract infection because older patients have a cold, right? So what you generally see is GI related in an older population. You'll see, infections in an older population. The disease itself, makes people prone to infections as well because they're immunocompromised. So, and then obviously when you're using a subcutaneous injection, you might see a little bit of injection site reactions. But in general, I would say that the AE profile is pretty manageable.
I would say, broadly speaking, if you think about T-cell redirection, there are two buckets of AEs that are related to the mechanism of action. One is cytokine release syndrome, meaning through the activation of T cells in the process of them killing the B cell, in this particular case the B cell, it doesn't really matter, you release gamma interferon, and then what ends up happening is a cascade. Gamma interferon stimulates other cells to release other cytokines, and so you get this cytokine release, broadly speaking. And the symptoms of cytokine release are well understood and characterized. They are fever as the first one. If it gets a little bit more extreme, it can be fever and hypotension, or fever and a minimum requirement of oxygen. And then the grading follows that logic, to the degree that the hypertension becomes more difficult to manage, requiring potentially pressors, or the oxygen demand gets significant. That's the first bucket of AEs that are related to the mechanism of action. Epco is not different. The second bucket is a ill-understood phenomenon of neurological symptoms, subsumized under the term ICANS. And it's really ill-understood, really, and there's no good explanation except that some kind of hand-waving idea of that you activate T cells, presumably CD4s for this particular toxicity, and that they have some weird interaction that leads to confusion, transient usually, headaches. What we now summarize ICANS is not necessarily what we used to think of neurotoxicity. So it's basically now every term that falls under this bucket of the system organ class. So headaches, confusion, but it can be problematic to the point that people are really, unable to communicate in very severe cases. Not with Epco so far. So that's kind of broadly speaking. And then there's a unique third bucket, I would say, to the CD20 targeting, which is neutropenia, which is probably real as well, which is true for rituximab as well. And all it says is that there's some biology that we don't really understand between CD20 positive cells and neutrophils. These kind of like the three buckets. So there's in that range, Epco behaves like the other ones in the class. There's really nothing to the subcutaneous administration, and I want to correct you. It's not really formulated. Rituximab was formulated to be, it was basically a chemical modification. It used an enzyme to allow it to be administered subcutaneous. It's very similar to daratumumab in its subcutaneous infusion. With Epco, it's purely the administration of subcu. The antibody is in the same solution as an antibody that would be given IV. It is just so potent that the amount of protein that you need to give to a patient is relatively limited. For Epco, it's 48 milligram total that you can actually administer that small amount in a very small volume under the skin, and it gets absorbed really well.
Yeah, I think I would just reiterate that what we see is really class effects across either anti-CD20 or T-cell engagers. And we've been developing Epcoritamab to be kind of patient-friendly. And really using the step-up doses to mitigate any grade 3 or 4 CRS or ICANS that we might see. So there is a bit of intelligent trial design to mitigate, in-class effects.
It sounds great. And to just add on to that, could you just describe the trial design?
The current data that is leading has led to the registration of of epcoritamab in diffuse large B-cell, and also the data set that is now being submitted to global health authorities in follicular lymphoma comes out of an expansion cohort of the original first-in-human trial. So the first-in-human trial was expanding the doses, and then once it's settled on the recommended phase 2 dose, it went into distinct populations that would meet criteria for an unmet medical need. For diffuse large B-cell, this is patients who had 2 prior lines of therapy and are not eligible for transplantation. And for follicular lymphoma, this was patients who had also 2 prior lines of therapy. So third-line patients in both settings, broadly understood as patients for which there is no really well-defined standard of care. What Jacqueline was referring to is that the initial step-up dosing, all of these drugs use a step-up dosing, meaning they start at relatively low doses and then in 1 or 2 steps get to the recommended dose to get the full effect of the drug, and that is meant to mitigate the cytokine release. That in a subsequent iteration of the study, we also generated data that helped us to understand how to further improve the safety profile, such that we significantly were able to reduce the cytokine release profile and completely abrogate any evidence of ICANS. And that is through to particular PK modeling that allowed us to understand that in follicular lymphomas, for example, specifically, a second step up introduced, basically a more gradual increase over a period of time would actually really fundamentally change the safety profile, such that at this point now the drug has mainly grade 1 CRS, which basically patients only have fever. There's a less than 10% rate of grade 2, meaning people who require some infusion of fluids, 500 cc's. Both of these things are easily managed in an outpatient setting. There's no grade 3, meaning there's no need to hospitalize, and there's also no ICANS, meaning there's no risk of potential neurologic toxicities with that step-up dosing. And that's very important because the ambition, of course, the aspiration for a drug from the very beginning, also because of the subQ administration, was that to provide an easily administrable medicine that can be provided to patients in all kinds of settings, not only in specialized hospitals.
That's great. And interestingly, in the phase 1 study, did you do inpatient admissions for the first few patients, just because that is becoming such a thing, with a lot of these bispecific antibodies and novel INK cell based therapies, that there's so many inpatient hospitalizations that's a unique aspect of this drug, that you don't require that, because it really is a logistical hurdle a lot of people are facing.
Yeah, so the reality is this: you have to dial the clock back a little bit. Right? So EPCO was started in 2018. It was actually the last of the 5 CD3 CD20s that went to the clinic. The first CD3 CD20s that went into the clinic were in end of 2015. At that point, the knowledge base, broadly speaking, on how to safely manage this set of symptoms that we spoke about, CRS and ICANS, was very limited. All experience came from CARTs, where these symptoms are significantly more profound. So cytokine release syndrome is much more severe. ICANS is much more severe. And so for these very reasons, essentially everyone was mandated by health authorities for appropriate reasons to require hospitalization to provide appropriate observation, until we started to understand how to better manage, which I think is fair that the community in general has learned this, to better manage this. And also, as we just alluded to, understood how to better mitigate it through the administration of fluids and steroids, and for example, diffuse large B-cell, and through the modification to the step-up dosing. And so now you're getting to a situation where the rate is lower, the severity is lower, the experience in how to manage this is better, so we better understand how to manage it, and so it becomes much more realistic to apply this safely, because in the end, patient safety is the first one. But I understand, from a healthcare resource point of view, it's a very important question to be answered.
Perfect. The short answer to your question is yes. We always try to hospitalize when we don't understand what the safety profile is going to look like. But I think the team has done a lot of work moving into a pre-regulatory state to get us to a place where hopefully we can bring this molecule in a completely outpatient setting in the future.
Would you like to touch upon some of the major study findings and how that would compare to some of the other T-cell engagers?
In particular, the NHL-1 study had lots of arms. We've already used the DLBCL cohort, LBCL cohort for our current approval. And what we're I think discussing today is the follicular lymphoma cohort. Patients in this cohort were very heavily pretreated. We use the term POD 24 to patients who progress off of chemotherapy and within 24 months. So in follicular lymphoma, generally patients get 5-6 years median after their first therapy. This cohort of patients was pretty heavily enriched with patients who were either refractory to their first therapy, refractory to the 2 prior lines of therapy, or progressed within 2 years, which is, they're all kind of markers of maybe less indolent follicular lymphoma. So these patients were in need of something a little bit stronger, which is why obviously they opted to go on a clinical trial. In that population, we were able to achieve an 82% overall response rate with 63% CRs. In follicular lymphoma, obviously it's an indolent disease, and you don't aim for you aim for durability, but CRs are a very good predictor of that. And so we're very, very encouraged by the rapidity of response. You can see median time to CR is 1.5 months, which is really driven by the first restaging of patients. And between that and the step-up dosing, we really think we have something strong we can offer patients, but also in a way that it can be administered across practice settings. In terms of comparisons to the field, as Tahi stated, there's no real standard of care in follicular lymphoma, particularly third line, and particularly for patients with this level of aggressiveness in their disease. So across the practice care settings, you can reach for potentially a bispecific or another T-cell engager. Those come with some, in particular, the CAR Ts come with a lot of logistical challenges, and they're not available to a lot of patients. There's also a fair amount of limitations to where you can access CAR Ts in the space. And a lot of patients with an indolent disease don't really want to opt for the level of hospitalization and rigor that is required for moving towards a T-cell engager. So I think we have a pretty good option for really deep remissions in some of the most aggressive phenotypes of this disease, and moving forward, potentially all lymphoma patients.
Yeah, and I think the only thing to add is, like, if you think about the class of CD3, CD20s, like in follicular lymphoma, they, from an efficacy point of view, they tend to behave very similar. That probably is a reflection of the functionality of T cells in follicular lymphoma, to be honest. And diffuse large B cells, it's a little bit more easier to see differences between the constructs, right? So there you can see, if you look at the data for all 4, Epcoritamab has the highest response rate and then the higher CR rate in the cross comparison. This is always dangerous cross comparisons. In follicular lymphoma, they are more or less the same, but here I think it's important to note that with the optimization, the safety profile of Epcoritamab is extremely competitive. It's actually the lowest rate of grade 2 and above for all of them. And it's the only one at this point that is administered subcutaneously, which we discussed before, has significant advantages in terms of convenience for patients. Right? And it's basically a difference between several hours in whatever setting versus a minute. Well, it's never a minute because you have to go to the provider, but nonetheless, it's a significantly more convenient way of doing things.
No, it's a great point. I mean, at the ASCO Quality Care Symposium, there was several sessions focused on time toxicity to patients.
Yeah,
so infusional drugs versus subcutaneous drugs. I mean, there's a lot of quantifiable ways it's better for patients. And on infusion centers, less cost of staff, less cost of administration. Those are phenomenal results in a heavily pretreated population. Any plans for further expansion cohorts in other disease states?
I think we've seen we've seen proof of concept in most B-cell malignancies. Obviously, we have a third line approval in DLBCL. We're moving into second line and front line with combination therapy, which we're very, very excited about, and trying to improve on cure rates in DLBCL. For follicular lymphoma, we similarly are moving into earlier lines of therapy with combinations to try to deepen and prolong those first remissions. And there's a lot, we just released our data in CLL this year at IWCLL, very exciting data and very heavily pretreated patients. CLL has been a disease state that I've spent a lot of time in. So very excited to have options for patients that where there really are no options in the BTKI BCL2 progressor or AA exposed patients. But that's probably not the only place we're gonna land in CLL as well, considering it's well, in my opinion, a better anti-CD20, so it could go wherever anti-CD20s are.
I think, like, broadly speaking, right? I mean, I think the promise of this new class that is emerging, and I think about this as a class, of course, initially, is really it combines the unprecedented, and it's really, and the term unprecedented sometimes gets overused, but these efficacy results for T-cell engagement, generally speaking. Now we're speaking about lymphoma. We could talk about myeloma too, by the way. But the efficacy results for T-cell engagement are without a precedent. There's never been drugs before that had like an 80%, 60% CR. This is like, unheard of as a single agent in refractory patients. But the promise of this class is that you can actually combine it, in contrast to CAR-Ts. You can combine it with other modalities. You can combine it with lenalidomide, which we're doing. You can combine it with chemotherapy, which we're doing. For different reasons, right? You can combine it with chemotherapy because chemotherapy debulks and now you have a better immune control. You can combine it with lenalidomide because lenalidomide probably in some weird way enhances T-cell function, and then you get synergism, and we have shown that in the clinic already. And as you combine it, you can move it into the earlier settings. And the ambition, the goal has to be to move this mechanism into frontline. And so together with AbbVie, I think we're working on what is a very aggressive, very comprehensive clinical development, moving this very systematically along the lines. We have already a phase 3 ongoing in the relapse setting for diffuse large B-cell. We have a phase 3 ongoing in frontline in combination with chemotherapy with R-CHOP and diffuse large B-cell. We just announced that we are going to start working also with collaborative study groups on doing a combination in frontline for patients with diffuse large B-cell who are elderly, who are not candidates for full R-CHOP. We're generating data for cardiovascular impaired patients who are really not candidates for anthracycline in diffuse large B-cell frontline. And also, we announced that we're going to do like a study in relapse setting with lenalidomide, really comprehensively addressing all spaces of diffuse large B-cell. Similarly, in follicular lymphoma, there's already an ongoing study in the relapse setting with lenalidomide and rituximab, with data that we had presented that was really unprecedented. With now in combination, CR rates way over 90%, like 98%, which basically means that one patient on your study didn't respond. It's like, there's like 100, it's like finite. Sometimes people misunderstand this. Like, you cannot go higher than 100. And now we are generating data in frontline and in follicular lymphoma, and I think Jackie already mentioned that we also have data in CLL, the same kind of, broadly speaking, it's a CD20 targeted mechanism. It has a play in all diseases that are CD20 positive, and there's a practicality to how drugs being developed and the time it takes. It doesn't happen overnight. But the ambition is clearly to position EPCO as a main mechanism across the spectrum of B-cell malignancies and across all lines.
And I would be remiss if I didn't say that it's EPCORE core therapy. That's where the team is trying to set the tone.
One last question, if you have the time for it. I was wondering if there's any major exclusion criteria that you had in your trials, or any patients that cannot be considered a candidate for this therapy in terms of their comorbidities.
Well, I mean, they're no direct. I mean, they obviously always, every trial has inclusion exclusion criteria. Usually in clinical trials, they're around performance status. Otherwise, I think the only other exclusion criteria that's probably worth mentioning is that because for unclear impact on the safety, we initially excluded patients with prior allo transplant. This is something that I think we're now exploring very gradually, where we now allow patients to come on studies who had a prior allo transplant, but they have no existing graft versus host that requires immune suppression. So, beyond that, I think well, actually one of the main differentiating aspects of these bispecifics is that they are available and can be administered to patients who may otherwise not be candidates for CAR-Ts because of their frailty or other preconditions that preclude them from getting involved on CAR trials or getting access to CAR trials. So, I mean, as you cross compare, that's true for all bispecifics, by the way. The patients who came onto these trials, in many cases, were either not eligible for CAR-Ts, or in about half of the patients actually had prior CAR-Ts. And so that's also the context in which one has to understand as we cross compare data. It's a significantly more pre-treated, a significantly more frail patient population than the CAR therapies included in the clinical trials. And I think this is the promise, that this is truly a much more egalitarian way of approaching the disease than a scientifically very interesting, but nonetheless very exclusive approach that can only be provided in very specialized centers under specialized care and to hand-selected patients.
That's very exciting. And actually, I was listening to another lecture on DLBCL yesterday, and there were some debates about whether some of the bispecifics can be used as a bridge to CAR-T. Do you think that that's also a possibility for EPCORE?
Sure. I mean, there are going to be all kinds of investigations in the community to figure out how to use these mechanisms for whichever practice you choose to do. I personally would say that's probably not the long term. I don't think that's going to be the place for the drug. The general philosophy of drug development, that you provide optionalities so that physicians and patients have different kind of opportunities to benefit from novel therapies.
I mean, I think to Tahi's point, over time, we would hope our combination therapies can stand up to the long-term outcomes that we see with CAR-T. I know to quote Tahi, there are people who they believe in the therapy because they've been around long enough to demonstrate outcomes that are predictive by the outcomes that we show. But we need a little bit more time to demonstrate their ability.
Perfect. Well, thank you both for joining us today. This is really interesting stuff to talk about, and obviously phenomenal results and a patient population that really needs a new therapy, and especially the subcutaneous delivery mechanism is, you can't underscore that enough. I mean, that's huge for patients, and that's going to make things easier for infusion centers too, and it's really going to make it more applicable to centers all over the world, as opposed to just in large academic medical centers where they're more trained to deal with these things. So very exciting stuff, and thank you both so much for joining us.
You're welcome.
Thank you.
Thanks.
Very nice to meet you.